In this video, we will discuss about this pancreatic cancer TME design, or in other words, we will continue our discussion about this pancreatic cancer tumor microenvironment design. So, in the previous video, we have understood that they have used 1:10 mixture of this KPC with tumor cells and pancreatic stellate cells to inject into the mice, and then once the tumors reach to this 1000 mm size, they analyze those tumors. For different CF markers. So in this panel I, what they have did presented the transcriptomic analysis of this human group and compare it with this 1:10 mixture for different cancer associated fibroblast markers. And what they are able to find out that in this 1:10 mixture, the CA associated genes are overexpressed or more expressed compared to the human group. This is evident from the transcriptomic analysis. So now they wanted to check. So this see a slight difference. However, they wanted to check whether there is a difference between human TME and 1:10 mixture tumor based TME. What they did is they did the PCA plot analysis. In the PCA plot analysis, they are able to see that this human cells or human groups, samples from the human groups and samples from the 1:10 mixture forms a single cluster where the cells from 1:10 mixture are tightly regulated. So this indicates that this tumor microenvironment design is the tumor microenvironment design from 1:10 mixture and the human group are similar to each other. So afterwards, they also checked this cancer associated fibroblast scores. In the case of the transcriptomic analysis, we see that the CA genes are slightly overexpressed or are more expressed compared to the humans. However, when they check the CAF scores, they are able to see that this 1:10 mixture or cancer associated fibroblast from 1:10 mixture and the cancer associated fibroblast from human group are similar to each other. That is, there is no statistical difference that they are able to find out, or it is not mentioned. So from this result and the results that they presented before, they are able to make a point that tumor microenvironment design that they have done by using 1:10 mixture of KPC cells and pancreatic stellate cells into the tumor mice seems to replicate the TME, which is rich in carbohydrate, rich in this fibroblast. So they can use this TME to check the effect of this oncolytic herpes simplex virus on this TME, as the literature says that, before this paper, the literature says that this oncolytic herpes simplex virus affect TME in a way that it will suppress the tumor progression. However, what are the mechanisms behind this functioning, or what is the relationship or mechanisms by which this oncolytic herpes simplex helps the immune system to come and detect the tumors is not been known. So for that, the initial step is to develop a TME, this tumor microenvironment, that can replicate TME similar to the human group. So which is essential in order to study the effect of this herpes simplex virus or oncolytic herpes simplex virus on this TME. From the first figure, they are able to come to a point that the TME design is close to this human group, and they can use this TME design for their further studies on oncolytic herpes simplex. So that's all for this video. I might have repeated some things twice so that you are able to understand it clearly. Even if after that, if you have any doubt, you can write in the comment or go through the reference where you can go through the detailed results descriptions and even if you're not able to understand any of the things, you can write in the comment box. So that's all for this video. Thank you for your time.